In this video, I'm going to present about the plant that we have studied, which is Catharanthus Roseus. Catharanthus Roseus is popularly known as Madagascar periwinkle. The characteristic of Catharanthus Roseus, which is it has a very good tolerance against heat, glossy, dark green, and have flowers in the hot weather. Catharanthus Roseus is a very important medicinal plant, belongs to Apocynaceae family. Their main metabolites, terpenoid indole alkaloids, TIAs, are widely used to treat human disease, and many of the terpenoid indole alkaloids are anti-cancer agents, which include azithromycin, catharanthine, serpentine, vindoline, vinblastine, and vincristine. In this video, we will focus on vincristine. The most important alkaloids in the Catharanthus Roseus, which is vincristine, that are used to fight cancer, which is derived from leaves and exhibit anti-cancer and anti-diabetic property. This is the chemical structure of vincristine. Firstly, this slide will focus on how to collect Catharanthus Roseus. Before start collecting plant, we need to get permission from local authority where we want to collect the plant. Before start plant collection, some precautions during plant collection, which is wearing field clothes and cover head to toe if collecting is to be in cold of winter or when mosquitoes or flies are in abundance. All the equipment were complete, such as plastic used to store the plant, cutter, tagging to label plant sample, and logbook to record all the data obtained. From one journal that we have studied, the fresh leaves of Catharanthus Roseus, which is purple variety, were collected freshly from the Garden of Almond Pharmacy Institute, OPI, Al-Watiah, Muscat, the Sultanate of Oman, to use for analysis. For the storage of the plant leaves were washed with tap water to remove soil and unwanted dust particles. From the journal study, the leaves were shaded, dried at room temperature, and then powdered using mechanical blender and stored in a sample container using for next analysis. After that, the sample must be vacuumed so that no air and moisture in the sample container. The sample container must be stored at low temperature, about -10 degrees C. Watering is very important because it involves keeping detailed recorded information about the collecting site locations, soil conditions, ecological habitat, date of collection, plant identity, and who collected the plants. In the journal that was studied, the leaves of Catharanthus Roseus were collected locally from the Garden of Oman Pharmacy Institute, OPI, Al-Watiah, Muscat, the Sultanate of Oman in September 2016. The plant was identified by the plant taxonomist and curator from the Faculty of Science, University of Alexandria. Butcher specimens of the plant's material were deposited in OPI specimen number Catharanthus Roseus 9.16. The next important thing is plant identification, which is we need to have a botanist in the team when do the scientific expedition to identify plant species based on their morphology. From the journal study, morphology characteristic of Catharanthus Roseus is an evergreen subherb or herbaceous plant growing to 1 meter tall. The leaves are oval to oblong, which is 2.5 cm to 9.0 cm long, and 1.0 cm to 3.5 cm broad glossy green hairless with a pale midrib, and short petiole about 1.0 cm to 1.8 cm.
As of right now, I will explain extraction of vincristine from Catharanthus Roseus. From the journal study from this assignment, supercritical fluid extraction was used for extraction vincristine from Catharanthus Roseus. Every extraction 0.5 gram of plant and powder was mixed with 1 mm diameter glass bed and placed in 3 ml stainless steel vessel. Ethanol as co-solvent was added onto the sample in extraction cell. The flow rate of supercritical carbon dioxide was nearly 0.3 plus minus 0.05 ml per minute. The extraction process was run, the vials was put at the room temperature. After supplementation of each run, the extraction yield was calculated. Here I attach schematic instrument setup of extraction. I will show that every component in schematic instrument setup of extraction. The first component is carbon dioxide cylinder. The second one is condenser. In the condenser there is shrink pump is located. Next, heat exchanger, then followed by oven. The extraction cell is located in the oven. The seventh component is restrictor, and lastly collection chamber. Optimization of supercritical fluid extraction experimental condition. In this section, I will explain how to optimize condition of supercritical fluid extraction. Firstly, we need to select carbon dioxide modifier which has an intense effect on the extraction yield. To investigate the extraction yield of Catharanthus Roseus by using different organic modifiers such as ethanol, methanol, acetonitrile, and hexane. The other experimental parameters were pressure at 350 atm, extracting temperature 40 degrees Celsius. Besides, the time of static and dynamic extraction 10 minutes and 45 minutes respectively. Lastly, modifier volume 100 microliter. Based on the journal that I studied, ethanol was chosen as carbon dioxide modifier because of the extraction yield was higher than the other modifier. Afterward, the other factors that impressed extraction yield were selected and optimized to determine the best condition in the supercritical fluid extraction process. The result was obtained from table 1. I want to highlight that the highest percentage yield is 7.5% at 200 atm, 20 minutes and 150 microliter. However, the lowest percentage yield is 0.5% at 260 atm, 37.5 minutes and 187.499 microliter. Therefore, the optimum point was observed at a pressure of 159.02 atm, time of 8.068 minutes and volume of 167.504 microliter.
After sample collection, vouchering and extraction, we will proceed into the next step, which is isolation of natural product. The components in the previous extraction are complex, containing a variety of natural products that must be separated and purified to achieve the active fraction of pure natural products. In isolation process, several types of separation techniques can be done based on its properties, which are: firstly is the adsorption properties. Because of the simplicity, high capacity, and low cost of adsorbents such as silica gel and macroporous resins, adsorption column chromatography is frequently employed for the separation of natural products, especially in the first separation stage. The separation is based on differences in the natural product's adsorption affinities for the adsorbent's surfaces. Secondly is partition coefficient, the liquid-liquid extraction principle, which is based on relative solubility in two separate immiscible liquids, is used in partition chromatography. In the beginning, one liquid phase was used as the stationary phase and another liquid phase was used as the mobile phase on a solid matrix such as silica gel, carbon, cellulose, and etc. Commercially available alkyl, aryl, cyano, and amino substituted silanes are frequently utilized as bonded phases to separate a range of natural products, particularly in the final purification step. Thirdly is the molecular size. The molecular sizes of natural products are used to separate them using membrane filtration, which is MF, or gel filtration chromatography, GFC. Next is the ionic strength. Ion exchange chromatography (IEC) separates molecules based on their net surface charge differences. IEC may be used to separate some natural compounds such as alkaloids and organic acids that have a functional group capable of ionization. For the separation of alkaloids, cation ion exchange resins were utilized, whereas anion ion exchange resins were used for the separation of natural organic acids and phenols. And lastly is the other modern separation techniques, which are molecular distillation, preparative gas chromatography, supercritical fluid chromatography, molecular imprinted chromatography, simulated moving back chromatography, and multidimensional chromatographic separations. In this step, Vincristine in Catharanthus Roseus will be further detected and determined using reverse phase high performance liquid chromatography, which is RPHPLC, which complement both qualitative and quantitative analysis. HPLC is a powerful and adaptable chromatographic technology for separating natural products in complex matrices such as crude extracts for selective detection and quantification or general profiling. The approach is widely used and has been developed to analyze a wide spectrum of natural products without requiring complicated sample preparation. Because of the diversity of natural products and the lack of a single strategy for their efficient detection, selecting the proper detection method in HPLC is critical. As referred from the journal, Shimatsu Model 10AVLC equipped with binary delivery pump model LC10AV is used to analyze standard material and samples. UV visible detector of SPD20A provides detection of the eluted peaks. Further details on the separation condition for Vincristine analysis is listed as follows. Next, preparation of standard solution will be done in order for the analysis to happen after considering the conditions stated previously. Dilution of the original pure standard Vincristine 1 mg per milliliter yielded 2 g per milliliter. In the HPLC, 20 microliter of standard was injected. The results were then shown in a chromatogram which contained the peak top, retention time as well as the area under the peak. After that, detection and estimation of Vincristine in Catharanthus Roseus were done by aqueous extraction, also using HPLC. The procedures are as follows. From HPLC technique that has been done, calculations were made to determine the concentration of Vincristine in aqueous extract as well as in original plant. As per journal recorded, the concentration of Vincristine in aqueous extract is 6.168 microgram per milliliter, while the concentration of Vincristine in original plant is 148.032 microgram per gram. The concentrations of Vincristine that has been calculated shows that quantitative analysis is successfully achieved. Meanwhile, for the qualitative analysis, the appearance of chromatogram peak for standard material as well as sample shows a very close retention time, which are 13.919 minutes and 14.068 minutes respectively. These values indicate that both compounds have the similar molecular weight, physical and chemical properties.
Characterization of natural products is significant to identify whether the compounds of interest are present in the sample plant. As we recall, Vincristine from Catharanthus Roseus is classified as an alkaloid. Qualitative analysis using spectroscopic technique, specifically infrared IR spectroscopy, can be carried out as the method of characterizing the alkaloid in the sample plant. From one of the journals studied for this assignment, characterization of Vincristine from Catharanthus Roseus is analyzed using IR spectrophotometer and FTIR. The difference these two makes is that IR spectrophotometer is a standard instrument. FTIR introduces a minimal sample preparation prior to analysis, as well as all form of sample can be analyzed. The first part is by using IR spectrophotometer. Prior to analysis, two separate transparent discs are prepared. One is the standard VCR and one for the sample, which is dry grinded. Each of them is grinded with potassium bromide before being compressed by piston under high pressure to form the transparent disc. This is the data of VCR standard presented from the instrument, while this is the sample containing vincristine. From the IR spectrum of sample, NH can be seen at 3419, CH stretching at 2926, C double bond O stretching at 1649, CN stretching at 1028, and double bond CH bending at 675. However, it is difficult to distinguish the functional groups because the sample spectrum does not look identical to that of standard spectrum. The dissimilarity might be resulted from low content of vincristine in the sample compared to the standard vincristine. The other instrument is by using FTIR instrument. Some changes have been made. One of them is that samples are directly placed on the accessory crystal for examination. There are many advantages of using advanced FTIR instrument. One is the spectrum of sample is almost identical to standard spectrum. Number two, more functional groups can be distinguished to compare the standards and the sample. And number three is practical for any form of sample such as liquid, solid, and etc. This picture shows the spectrum of standard vincristine, and this picture shows the spectrum of sample. NH can be seen at 3298, CH stretching at 2922, C double bond O stretching at 1629, C stretching for aromatic ring at 4019, CN stretching at 1029-25, and double bond CH bending at 668. From both instruments used, the presence of vincristine in sample of Catharanthus Roseus can be approved when compared to respective standard. These are the overall bonds that present in the sample. They correspond to the bonds that present in standard vincristine. This table summarizes the comparison of wave number of standard and sample from each spectrum of each instrument. We get to learn much more about extraction and isolation of natural products, especially vincristine from Catharanthus Roseus, despite knowing the benefits that the compound offers to our health as a drug to treat cancer primarily. It is important to have the knowledge first before collecting the plant sample from a certain place. Extraction process using supercritical fluid extraction to extract vincristine from the leaves of the plants collected prior to isolation and purification of sample using chromatography approach, which is high performance liquid chromatography, HPLC. The final step is by using spectroscopic technique, which is infrared spectroscopy. This is to characterize vincristine from the sample as well as to verify the existence of vincristine from Catharanthus Roseus leaf sample. These steps are the fundamental of the development of drug, which is beneficial for the use of medical, pharmaceutical and alternative medicine.